Hi there, my name is Morgan Ellis, and today I'll be talking about how we used eDNA in detecting two invasive species along the coastline of Victoria. First of all, I would like to acknowledge the traditional owners of the land I live and work, the Wurrung and the Bunurong people of the Kulin Nation, and pay my respects to their elders past, present, and emerging. Port Phillip Bay is one of the most invaded bays around the world, with approximately 180 introduced species. This doesn't come as a huge surprise, as it contains the city of Melbourne and the port of Melbourne, which is one of the largest ports in the Southern Hemisphere, that sees a lot of international shipping moving in and out. International shipping, and in particular the ballast water, being a major vector for transmission of invasive species. We all know invasive species can have a large environmental and economic impact, so being able to detect them early and jump on them quickly is really important to sort of stopping that damage. There's also a lot of domestic shipping, including recreational vessels that move in and out of Port Phillip Bay. So there's a risk of these spreading from Port Phillip Bay along the Victorian coastline. One key invasive species in Port Phillip Bay is the Northern Pacific sea star, Asterias amurensis. As you can see, it's native to areas of Japan, Korea, China, and Russia, and Alaska. It was introduced down to Tasmania via the ballast water of ships in around 1986. It was originally misidentified as being a native species, and in that time it spread up to Port Phillip Bay in around 1996. And one female can release 20 million eggs at a time, so it didn't take long for this to become a huge problem, with an estimated population in Port Phillip Bay of 165 million sea stars by the year 2000. It looks something like this. The North Pacific sea star is a hugely voracious predator. It'll eat basically anything it can get its hands and arms on. So it just decimated local bivalve populations. It has since spread to Europe, the Americas, and Australasia. The key vector here is, again, commercial and recreational vessels. It's often found biofouled along their hulls. Wakame is a highly invasive algal species. It has rapid growth and high fecundity, which allows it to spread and grow quite quickly. Through this, it's able to displace native species of algae, and through that, undergo major ecosystem transformations, disrupting all of the species that are then reliant on those native algals. It also has this planktonic life stage where the adult plant will die back, leaving just the plankton in the water, and this makes it quite difficult to detect at certain times of the year. Both Wakame and the Northern Pacific sea star have had various incursions along the Victorian coastline, with eradication efforts having varied and sometimes limited success. For example, the Northern Pacific sea star was thought to be eradicated from Wilson's Prom, which I'll show you on the map shortly. However, since then, a new population has been detected there. This is obviously a concern for marine protected areas as well as ports and harbours all along the Victorian coast. So how do you detect an invasive species? Traditionally, this would involve things such as diver searches, time surveys, and drop cameras. As you can imagine, this is quite time consuming. It's quite expensive. You often need 2 or 3 divers in the water, as well as people on the boat, plus the boat use, fuel, the cost starts to rack up. You also often need specialist knowledge to be able to identify cryptic species, and it's easy for a diver or a drop cam to miss small numbers of invasive species that haven't quite grown into an established population, which is really when you need to be getting on top of them. There's also, as you can imagine, a lot of OHS issues in trying to get divers and boats in and around high energy rocky shorelines. This is where eDNA can help fill this gap. Using environmental DNA is relatively cheap and time efficient. It can also detect small populations in minute quantities of DNA and also be able to separate cryptic species from native species. This involves taking a small water sample, using something such as a Sterovix filter to filter out the DNA, extracting that DNA and analyzing it. And there's two main approaches for analyzing it, using DNA metabarcoding, which will detect a wide variety of things in the water, but you need a higher quantity of DNA, or a species-specific approach, which is what we've used in this case study here, which is able to detect quite a minute amount of DNA, but it's specific to the invasive species that you're looking for. qPCR approach uses species-specific primers and probes, and they're amplified on a qPCR. We take our environmental sample, we extract the DNA from there and amplify it. And this is able to detect minute concentrations, often less than 10 copies per microliter. eDNA will decay over time in the water column, and this can lead to possible false negatives. The decay rate's influenced by both biotic and abiotic factors, including the type of species that you're detecting. Things like plant species that have a cell wall are often able to keep the cellular material together compared to an animal species. The cell will break apart quicker. It's also influenced by the UV, salinity, and one of the main ones is the microbial activity. This study focused on two invasive species, wakame and the northern Pacific sea star. We wanted to determine the rate that the eDNA decayed, determine the sensitivity of our assays, and then use this information to detect the species presence in Victorian ports and harbours and marine protected areas. eDNA in the water or not. This is done by using 5 serial dilutions and 12 replicates of each dilution. So a limit of quantification for wakame was 77 copies per microliter, and for the Northern Pacific sea star was 4 copies per microliter. And what's more important from a biosecurity perspective is the limit of detection. For wakame, it was 2.6 copies per microliter, and the Northern Pacific sea star was 1.4. So you can see these assays are quite sensitive, so they're able to detect quite low concentrations of DNA. From there, we wanted to determine our eDNA decay rates. So these were tested under laboratory conditions. So these should be viewed as a maximum decay rate, not a standard environmental decay rate. We took water samples from known populations and then filtered these at a regular interval over about a week-long period and quantified the amount of DNA on a qPCR, and then modeled these decay rates with an exponential curve. So you can see here the two decay rates from the two species, despite one being plant-based and having that cell wall and one being an animal without the cell wall, the two decay rates are quite similar. The majority of the eDNA decaying over the first 48 hours, then up to around about 90 hours, the decay rate slowed down, and after 90 hours, it dropped below limits of detection. So from there, we wanted to determine the invasive distribution around Victoria. So myself and my colleagues at Clark went to 14 locations, collected our eDNA samples, and then tested these for wakame and the Northern Pacific sea star. We ran each sample as 3 technical replicates. If we had 2 detections, this was a positive hit. If we had 1 detection, this was an inconclusive hit, which means some further work's needed to determine whether we trust the amplification or not, and 0 hits meant this was a negative detection. So you can see from this map of the Victorian coastline that we've got quite a few positive and inconclusive detections for both of our species. In areas such as Point Lonsdale here, it's right at the mouth of Port Phillip Bay. There's a lot of both Northern Pacific sea stars and wakame in here. So it's possible that these detections were from the water movement in and out of Port Phillip Bay while we were sampling. However, in areas such as Wilson's Prom down here, it was quite concerning to see both species, in particular the Northern Pacific sea star, reoccurring in some of these spots. From there, we sequenced the subsample of our DNA, and this is just to make sure that what we're seeing coming up on the qPCR as amplification is actually amplifying the DNA belonging to these two species. So these were blasted against the NCBI databases, and all of our samples returned a match for either wakame or the Northern Pacific sea star. This really provides confidence that what we're seeing come up as an eDNA hit is actually detecting the invasive species we're looking for. If you want to read more, this paper's been published in the Science of the Total Environment, so feel free to grab a copy and have a read of that. We also did some really cool biophysical modeling. I don't have time to go into that at the moment, but it's well worth the read. I'd like to thank everyone for their assistance, in particular my co-first author, Zach Clark, and I'd like to thank you all for listening. Thank you.